All right, so it's 5 past. I'm going to get started. We are recording the session, so will be made available to everybody once the webinar is finished. I do see some comments in the chat. Thank you very much. Please keep them coming. We like to hear a little bit about who you are and why you're here, so please do keep them coming. I will so long get started. So a very warm welcome to all of you. This is the challenge webinar for the second of two Mpeg-g challenges. This one referred to as decoding the dialogue. These two challenges are being run in partnership with um, Philips and with Mpeg-g. And we're very pleased to also be welcoming Viome as a data sharing partner, the partner for this challenge. So I'm going to get right into it. In terms of session expectations, please use the chat and use your reactions to participate. It makes it more interesting and more exciting for everybody on the call to see that you're engaged and listening. Please stay on mute, just in order to keep the recording clear and free of distractions. Stay on mute. Of course, if you'd like to ask a question, there certainly will be time for that. That'll be at the end. You're most welcome to ask questions. In fact, you're very strongly encouraged to ask questions. That's really why we run these webinars, so that you can engage with the experts that we have here on the call and you can get answers to your burning questions. But there will be time for that, and at that time you can feel free to come off mute, introduce yourself, and ask your question. If that's not practical, you can also ask questions via the chat. Thank you to those introducing themselves in the chat. Please keep it going. Let's please be respectful to others. And most importantly, please enjoy your time and please make sure you're engaging with the event as much as possible. As mentioned, we are recording the session that will be shared with all of you who signed up for the session today. So in terms of an agenda, we're right at the beginning with the introduction. That's what's happening now. I will go from here into a short description and explanation of the challenge, essentially an overview, and then we'll hand over to our experts. We have Nevenka Dimitrova from Philips, who will explain more about the origins of the study, the data collection, and so on. And then we have two experts from Viome, who will talk a little bit more about the data that's been provided to us and some exploration of the functional and technical aspects of transcriptomic data, which is the data that's being used for this challenge. That will be followed up by a Q&A. I will run the Q&A, but likely I won't be able to answer too many technical questions. Questions about the platform and about the challenge design, that will certainly be handled by me, but questions around Viome data will go to Dr. Lan or to Eric, and questions about the original study and more general questions around transcriptomics and metatranscriptomics can go to Nevenka. So that being said, I will get right into it. So this is our challenge description. The title of the challenge is Decoding the Dialogue, and the challenge really is to determine host microbiome interactions in metagenomic data. This is a challenge that builds on our first challenge that's currently also running on Zindi, our first challenge with Philips and Mpeg, which is more of a supervised machine learning challenge. This challenge is a little bit more open-ended, and so it will require more creativity and thinking from you. And we'll go into how the challenge is going to be, how you make submissions and how the challenge will be judged shortly. So your task will be to model and predict complex interactions between microbiome, taxa, and the immune system in various body sites in humans. And the idea is that there are some subtopics and you are being challenged to answer certain questions. This is, as I said, a report-based challenge. This is not a machine learning challenge in the way that other Zindi challenges are. In this challenge, you will be asked to submit a report. I'll go into this in a bit more detail, but your solutions are going to be judged based on a rubric rather than based on a single error metric. You're invited to compete in one or more of 5 different tracks, and each one will ask you to submit something slightly different. And we'll get into that shortly. And you can see some of our partners for this, the graphic on the right. So that is my cue to change screen. Please bear with me while I change over to a different screen. Stop sharing. Find the correct thing.
And is there a way to mute everyone else other than the speaker? It seems like we have some background.
I will do that now. Thanks,
Yes.
Anka.
Okay, great. Great. Thank you.
Okay, and get my screen again. There's okay. So you should now be seeing my screen once more. And we are now on the Mpeg-g decoding the dialogue palette on Zindi. Um, I'll pop in case we don't have it, I'll pop the URL in the chat, so you're welcome to follow along. So feel free to follow along, or of course, just follow along on my screen share. So as you can see, the competition launched about a month ago. We've got another little under 2 months to go. And there's a prize pool of 5,000 US dollars. This description is something we've gone through briefly already, so I won't go into too much detail. Nevenka will go through the original study by Zoe et al. And you can see a little bit more about the challenge here.
Can you
You're
zoom
being
in?
asked, yes,
Yeah.
it should be better. Okay, you've been
Okay.
asked to, it's okay?
Yes,
It's
thank you.
visible? Good. Um, yeah, I know I have a larger than usual screen. You've been asked to use a number of different modeling techniques like deep neural networks, temporal attention models, graph neural networks, and so on. And really, we're excited and interested in you bringing your own creativity to this challenge. So if you have some good ideas that we haven't mentioned, that's so much better. So here we have now the various tracks that you can participate in. The first track is cytokine level predictions from the microbiome data. Um, so as you can see, you are still required to be doing some model building. You just won't be making a submission to Zindi in terms of the predictions. We are still asking for machine learning techniques to be used. So using metagenomic profiles for body sites, you'll need to build a model to predict the cytokine profile for that scene. So we've given some recommended inputs, expected output, and here you have the goal. Minimize the regression metric of your choice across these files and include your RMC. I'll talk a little bit more about what your submission should look like for all of these tracks once we've gone through the tracks. The second track, track two, microbe cytokine association discovery. We're looking for associations between specific microbial taxa and related cytokines in the body. So here you will need to identify and rank important interactions similar to a Bayesian model. Your inputs are the microbiome and cytokine data, and your outputs should be a ranked list of microbe cytokine pairs with your confidence score. And the idea here is to match ground truth associations that we already have. Track three, graph-based host microbiome network discovery. This is a challenge to build a microcytokine interaction network. This is, I think, graph networking. This is something that we have, the original study has done, but not using machine learning. And here you have various inputs and outputs. So your output is a graph with nodes of microbes or cytokines and and you need to infer relationships between them as well as attack weights and so the goal is to match known or simulated associations, and we will giving bonus points for demonstrating biological plausibility. Track 4, latent health state discovery. So this would be identifying health states in the host with predicting, in fact, health state in the host with multi-omic data. And we're looking for you to use unsupervised or contrastive learning, and it should identify personal dynamics or immune phenotype. So again, input is microbiome and cytokine profiles over time, as well as metadata such as time point, health status, and demographics. And we are looking for embeddings per sample or per participant, as well as optional clustering or health states, keeping of health states. And the goal would be to reveal correlations with insulin resistance, disease progression, or other time-dependent events. We'll be giving bonus marks for visualizations with annotation. And then lastly, we have an open-ended discovery track. And this is really something that we want to encourage those of you who are feeling creative. We want to see what insights can be discovered from the data. We don't want to tell you entirely what to work on. So whatever tickles your fancy, if this is your area of study or you just find some interesting patterns in the data, this is entirely up to you. And some suggestions would be exploratory longitudinal data analysis, bioinformatics pipelines, methodological curiosities, unexpected relationships or trends, and benchmarking of Mpeg-g performance. So those are the tracks. When you are making submissions to Zindi, we have made it up to 50 submissions available. For each track, we're asking you to submit a zip file. The format is important for Zindi. We only accept certain file types. You're being asked to submit a zip file, which should contain your written report in PDF format, as well as your code. No matter which track you're submitting on, you need to include in your code and your written report, you need to include your methodology. You need to include your data processing and feature extraction. You need to include model architectures and your training strategy. You need to include the performance metric that you chose to use, the scores that are being output. We need you to include interpretation of results and insights. And then we need a small report on your runtime and resource efficiency. So that would be training and inference times at a minimum, as well as the specifications of your GPU or CPU. And we will take your most recent submission as your final submission. So you are welcome to make repeated submissions. The one closest to the close date of the challenge is the one that you will be scored. So in terms of evaluation, this is not an automatically evaluated challenge. This is going to be evaluated by a panel of expert judges, some of whom are on the call today. That is because of the nature of this challenge. It's not because it's quite open-ended and relying on insights and I guess some custom work. We want this to be judged by experts. We are placing an emphasis on technical merit as well as the potential impact of your submissions. And here is the rubric that we're using in terms of how to go about scoring your submissions. So 20% goes to scientific rigor, 20% goes to the performance of your model, accurate, interpretable, and or biologically relevant prediction, 20% towards innovation. So we want to see you going beyond the state of the art, not repeating work that's already been done in the space. 20% goes to communication. So that is, how is the report structured? Are your insights clearly communicated? Are your visualizations clear and well thought through? And then efficiency. As you saw, we've asked you to report on your specifications and your model efficiency. We want the models to be resource efficient and scalable. And you can see each of these 5 weighted equally. We have a bit more information on the page here about Mpeg-g. I'm not going to go into that in too much detail. Here we have prizes. We are issuing, sorry, it should actually be 1,000 per each track. That's an error on our part. That's going to be updated. That should be $1,000 per class. We are also really excited to introduce winners to the research team so that we could look into co-publishing papers or other perhaps conference proceedings or anything else that makes sense based on the siblings that you make. Okay, I'm going to pause there. I've definitely spoken enough. I'm now going to invite Nevenka to take over, and we're going to go into some of the technical details of the data that is available. Yeah.
Thank you, Paul. I just sent a sharing request.
Yes, you should be available to share now.
All right. Hello, everyone. Hello, 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 as we say. So this is very exciting. And I see how many people signed up for the hackathon and being active. It's really amazing to me to have so many people interested in such a fascinating topic, because it's really about the connection between the microbiome and health, meaning it is our environment and how we interact with our environment through our microbiome, and how this affects our health. I would like to first thank both Snyder Lab and Xin Zhao, now from Fudan University, for helping us with the data from the Snyder study. And later I will introduce Alan and Eric from Viome with a new data set, which is much richer, as you will see through their presentations. And I think we have already, Paul has already spoken about the fact that today we're focusing on challenge 2 and decoding the dialogue. And overall, we are really focusing on this interplay between what microbes are producing versus our response from our own immune system. In the Snyder study, we have this represented by the cytokine assay, meaning the immune system, and of course the microbiome data. And I just wanted to remind everyone about the overall Snyder Lab longitudinal microbiome study. So if you were not part of the first challenge, this is a 6-year study with 86 participants, and they have given samples, microbiome samples across gut, skin, nose, and mouth as body sites. And important thing here to note is, based on the Cell paper that they published, is that the most persistent bacteria were unique to each person, because this is 6 years and multiple samples per person. This kind of analysis was someone is talking. Can you please mute? And then what is important is that one of the focal points was people with type 2 diabetes. They showed less stable and diverse microbiome. And it's important to note that here in this massive, from the microbiome point of view and data that's in public domain, it's important to know that there are actually 5,400 samples, and that means something like 118 million data points that were analyzed and you have at your fingertips to analyze. The key finding was that the long-term multi-site monitoring revealed that the microbiome is actually fluid and a systemic ecosystem. Hello? Can you mute? Okay, thank you. So that's the high-level view. If we just visualize 86 participants, 4 body sites, and the important thing is that we have multi-omics data. We have proteomics data with the Luminex assay for the cytokine data, metabolomics, lipidomics, as well as microbiome data. Now, what's important is that they were able to predict insulin sensitivity versus insulin resistant individuals. And they were able to follow this reduced host microbiome interaction to the decreased gut microbiome diversity, and then that meant damaged skin microbiome and also a resistance to insulin. At the overall view of the study design, at the top panel, we see how it was envisioned, meaning every 3 months, every participant gave their own samples. And these samples were collected from blood, stool, skin, oral, and nasal sites. Then there is a data acquisition. So for proteomics, there are 302 samples, metabolomics, 724, lipidomics, 846 measurements. Cytokine, there are 62 cytokines, and there are 51 lab tests. Not all the data are available at all time points for all the participants, but the majority of the data is there. And very importantly, for the microbiome data, there are all of these samples, microbiome samples with 16S RNA technology that are collected over these 4 body sites. And the top right, as you can see, we have in just one figure representation of the number of samples. For lipidome, we have 1,440 samples. For oral microbiome, over 1,000 samples, and so on. For the cytokine data, as you can see, there are 965 samples. And then the same is done over the time points. So now you can see kind of the spread of the data. The first figure on the bottom right, bottom left, I should say, figure D is actually UMAP representation of the microbiome data, and that is really part of challenge 1. Now we will talk about challenge 2. More on challenge 2, I think, is in the paper. If you look at the paper, it's in figure 4, and there is plenty of description of the methods there. I think I already gave kind of a high-level overview, but the breakdown is really on the microbiome sequencing of these 5,432 microbiome samples. I think the important aspect here is that during events, there are additional samples that are being collected. So if someone had an infection or vaccination, or they were on antibiotics, they were collecting more samples than the usual 3-month sample. So that's why we should not assume that the data is all at set time points. The data is actually at set time points plus these events. Sorry about this. And this is one aspect that you can dive into, meaning what is the interaction between the microbiome and some of these cytokine data during these events. The cytokine data is really captured via multiplex assay, Luminex assay, and they were analyzed along the microbiome data. As you can see here, there is a link. We will share these slides. And there is a figure from the paper that I will try to also show. Maybe this is the right time. That's when I get to that. As usual, when you want, I know that it was right here. Yes, here we go. So this panel A represents between the cytokine data and the microbiome data. As you will see here, and I don't know if I can just zoom in a little bit. So this is really the mixed effects model outputs for cytokine microbe correlations. So the red lines are positive interactions and the blue lines are negative associations. And then each circle, the size actually denotes significant correlation count. So all of these unpronounceable names are the cytokines, and here all the representations for stool, oral, skin, and nasal genera from the microbiome study, as you will see here. So that's, in a nutshell, what the study tells us. There is also clinical and health metadata, because we have the dates also for these infection episodes, vaccinations, antibiotic treatments, and so on. And here, I would like to dive a little bit into what is a cytokine. I'm a computer scientist, so many years ago when I started learning and working in bioinformatics and biomedical informatics, I was Googling every single word. And some of this may be new to you, and I don't want to discourage you, but immunology books are one of the thickest books in medicine. But that should just encourage you to learn more. But on a very simple level, cytokines are signaling molecules of the immune system. A network of cytokines actually has this complex interplay of signaling molecules that regulate various cellular processes. So immunity, inflammation, hematopoiesis means how blood cells are generated in our body. And these molecules are basically messengers. They influence cell survival, growth, differentiation, and different effector functions. Eric will talk a little bit about this. And dysregulation of these networks can contribute to various diseases. And this includes autoimmune and neurological conditions. So on the right side, there is a very simple diagram. So there is cytokine gene, that's represented as a circle. There could be some external inducing stimulus, and this cell in the cytokine gene is producing cytokines, means proteins, that then interact with the receptor on a different cell. So this causes gene activation in the target cell, and then there are downstream biological effects. I have to redact myself. So the cytokine gene is not the circle, it's the cell in this figure. And what kind of cytokines are there that you will see in the paper? Intermediates. These actually, just based on the name, they act between leukocytes, between the white blood cells. But since they were named, now our knowledge has deepened, and it is known that they are produced by a variety of body cells. Interferons, these interfere with viral replication, and then there are colony stimulating factors, tumor necrosis factors, growth factors. There is a lot of them, and you don't need to go through all the details of learning the function of every single gene and their protein. For us, what's important is to focus on this fact how cytokine profiles and microbiome composition are intertwined, and they exhibit dynamic changes over time. So this can be influenced by disease, say insulin resistance, or viral infections, or lifestyle interventions, for example, food. And we already mentioned that in the Snyder data, there are 62 cytokines that are produced by so-called Luminex assay. So there have been multiple studies by Snyder Lab. It's not just the paper that we are focusing on. If you are interested, you can read more about it, but their research really highlights the connections between microbiome of different body sites, for example, gut, skin, oral, nasal, and then clinical indications, such as specific cytokine profiles and insulin sensitivity. One of their studies shows that interleukin-1, one of the cytokines, signaling strongly influenced microbiome abundance across all 4 sites, while 2 other molecules, 2 other proteins, leptin and GM-CFS, were more impactful on microbiome richness. So when you encounter these protein names, you can go and Google, and that's a little bit of deepening your knowledge, but please don't get stuck on just a single name. Just think of them as a group of molecules that interact, and this is what we are after in this challenge. Cytokine networks can look very complex, and much more complex than integrated circuits, I'm sure, at least in terms of meaning, terms of semantics, what each one of them does. In this diagram, there are different cell types, and what is depicted here is how these different cytokines are produced by which cell type and some of the interacting cells. And that's all we need to know on a very high level to start looking into the data. I hope this was helpful. I will now switch and introduce, we have Dr. Lan Hu from Viome, who is leading bioinformatics and data science at Viome, and Dr. Eric Patridge, who is leading computational systems biology at Viome, to introduce the Viome data. And then I hope we have some time for questions of how we can use both data sets in this challenge. Thank you. Lan, please, floor is all yours.
All right. Thanks, Nemanja, thanks, Paul, and thanks the organization to have us. Let me share my screen. Go to my presentation. Can you see my screen?
And we're currently seeing that you are trying to share your screen. There we go. It took a little while to load. It's now.
All right, cool. So let me get to it. Our data set is gonna thicken the plot a little bit, but by removing a strand, meaning our microbiome data is about RNA, it's not about DNA. So you've heard metagenomics, the terminology is quite a bit. And usually when people talk about microbiome, 99% of the time, it's about DNA and genomics. However, Viome, our company, we take the approach to sequence the RNA molecules from the biological samples. So, and I will explain why and why it could provide more real-time health and what's going on biologically or biochemically, the dynamics from the microbiome perspective, and such that it could also impact the host, meaning, us. So this is a very high level flow of how the RNA molecules can be extracted and sequenced. So we will collect our customers' biological samples, which I'm not going to disclose because it's part of the body site challenge you could think. And from the sample, the cells will be lysed at the collection. Then both DNA and RNA are present from those cells. But in our lab, we actually have a real clear certified, the wet lab, sequencing lab. The protocol would remove all these overly abundant RNA molecules, most of which are ribosomal RNAs. They're very abundant as part of a translational machinery. But then they're going to overwhelm the signal that we would like to measure. So here, the red squiggly lines are representing non-informative RNAs, and they will be mostly depleted through the probes. And then by doing that, we can enrich the green part of the molecules. Those are the interesting RNA molecules that can help all the downstream analyses. So after that, it's a typical multiplexing sequencing prep. And then we put on the sequencer, the machine is gonna run for a few days. And once that data is ready, it's pushed into the cloud. And our in-house built bioinformatics pipeline will kick in. And that, with various machine learning models and AI aspects, it will come in for the downstream work. So I'm going to just talk a little bit of this catalog, which is the reference universe for the bioinformatics pipeline to be able to identify what's there. It's typical for the sequencing data analysis, but we spent quite a bit of time to collect and curate what's the genome sequences, the gene information, annotation, and their biological functions, which Eric later gonna talk about. Because RNA, it's mostly messenger RNA, and they will be translated into proteins, which will do a lot of job inside the body. So that's why this part of the curation, it's a highly customized, and we keep updating. We would like to keep, as much updated as possible, but it's not everything. We also curated to make sure they're mostly validated. There are computational assemble the genome references, and the quality is good, then we will take into this collection. So this is the foundation of what this bioinformatics pipeline can see from a particular sample, right? So I'm gonna just describe briefly what our bioinformatics pipeline will be able to identify, and therefore the data you will be seeing. So this is a schematic illustration. Each gene, the color boxes, and the genome is a bar. And of course, we need to keep track of what genomes they are, which genes, and what genes belong to which genome. On top of that, this is the function part comes in. So all the bacteria, well, they may code slightly different version of a gene, but then in terms of the biological function, they will be doing the same thing. For example, alcohol dehydrogenase, we have it. A lot of organisms have it. The bacteria would have it, and a lot of enzymes that will help them to do all the work they need to do. So here I'm representing by these color bars. So the green, different shades of green from different bacteria genomes, for example, but they would be doing the same function. And then we also keep track of the taxonomy tree, because otherwise you wouldn't know what is the lineage, phylogenetic, the relationship of all these different genomes and how they are, in the hierarchical sense, this taxonomy rank. So we also have very extensive curation of the phylogenetic tree, taxonomy tree. Now, when a sample is sequenced, they will get a lot of these short reads. And the first step, any sequencing mapping tool would do, just map the reads to where it can be mapped from this whole collection. So here, for example, for these 3 genomes, I got these little black bar representing the reads. They can be mapped, wherever it can be. So the aggregation, the reads mapped to a gene, that will represent the abundance of the RNA expressed from this gene. And then all the genes map to a particular genome. These are collection of all these expressed RNA from this genome. So in a way, it could represent the abundance of this actively expressed this genome in a particular sample. And all these reads mapping to the same color group, those are all the expression of the same functional unit, so to speak. So that's our bioinformatics pipeline would do. And then later I'm going to describe a little bit more of the data format we are sharing, and you will have a better understanding of it. So for this challenge, we are providing 20 participants from a study, and it's longitudinal. Every participant would have 2 time points, roughly 3 months apart. This is baseline T2 is after some intervention. So 40 samples total, and then each sample would have 2 FASTQ, gzipped FASTQ files. It's 150 base pair long, the paradigm sequencing. So together you will have 80 FASTQ, gzipped FASTQ files. And we also remove the human reads as required by the challenge, because when we extract RNA, there are material coming from the human different locations wherever the sample is coming from. And then we also provide you the metadata description of what each participant, and I took a screenshot. Basically, it has a few columns. You have a subject ID, their age, their gender, their BMI, and their symptom severity level at time point 1 and also at time point 2. And these severity levels, there's one of these 4: healthy, mild, moderate to severe. So you could think of this as a label to describe the ground truth. And then the T1 to T2, these are their sample names. You would need to use this particular string to locate the particular FASTQ files. So for example, this is a sample name at T1, there's this long string, and you will find there are two FASTQ files with the no human, meaning the human reads removed, and this leading part, that's the sample name, and then there's something that's automatic added by the sequencer software. So that's your relationship to connect the various FASTQ files to a particular subject and the multiple time points. So that's the metadata and the FASTQ files. On top of that, we also provide you two long format tables, basically. One is about taxonomy abundance recount of every species identified in the sample. So I took a screenshot here just to show you. You will have a sample name, which is the same as the FASTQ name, and this is species name. The recount is the raw recount. You do your own to normalize however you wanted to process them. And then the following few columns, those are different taxonomy ranks. So there are genus, family, all the way up to phylum. I think this is compatible to the taxonomy classification outcome from 16S sequencing. So this, could bridge the data, the sequencing from the DNA or metagenomics based output and our metatranscriptomic based output. So this is one. And on top of that, this is our unique addition. These are the functions, meaning the biochemical functions, and we use the KEGG ortholog KOIDs to label them. Eric will talk more. And similarly, we'll have the sample name and this function name and the recount, also the raw recount from that particular sample, and what this KEGG ID means. Here, the K001 is alcohol dehydrogenase. It's just a little bit of annotation. So with that, I'm going to pass to Eric to talk about how those functions from metatranscriptomic data can be utilized.
Awesome. Thank you very much, Lon. I'll let you keep sharing if that's okay.
Yeah.
So as Lon said, I'm going to talk a little bit about the functions that we see and how we analyze the data set on our end. And most of what Lon talked about is upstream from half of this image that we're looking at. So the first two steps in this slide, sample molecular data and the genes mapped to microbial functions using reference database, are all encapsulated by what Lon was talking about. And a lot of what I'm going to be talking about is with regards to what we do with those mapped functions and how we analyze those with diseases and come up with signals that we can then quantify. And if you would like, I've also included a citation for a manuscript, which was actually accepted last week into BMC Gastroenterology. And yeah, so you can you can follow along here and then follow up afterwards and read the manuscript. So go ahead, next slide. So some of what we're talking about today is stool related. This particular slide is saliva related. And this slide is focused on karyogenic activity. So this would be activity from microbes in the mouth that causes cavities. And on this particular slide, we decided to highlight dyspepsia, diabetes, and Parkinson's as three different comorbidities, which are known to associate with cavities. I would say some of these might be causal and some of these might be an effect. In the center, we see a representation of the tooth and the gums, and then we see all sorts of molecules that are related to cavity formation, including acids, sugars, enzymes, and calcium. And the functions that express and modify and produce and eliminate all of these molecules is really where we spend our time. So you see on the bottom right, the cavity promotion, key components and key functions. These all represent things that have been defined by science that we can latch onto and say, this identifier, for example, K03332, is a fructan beta fructosidase. And these are some of the CAG or function identifiers that we can latch onto for trying to create signal as we walk through this data set. Now, on the top right are a number of different comorbidities that we identify in our data set. You can see the counts of people, and in the parentheses, n equals 8, for example, at the top, n equals 64 at the bottom. And all of these numbers that you see are how much differential we have between those people with that comorbidity and the people without that comorbidity. And that's on a scale of 0 to 100. So we're able to take specific functions and attach them through the bioinformatics process line described, and then select those and come up with a score. And I'm going to talk more about how we compose that score in following slides. Go ahead. So this is another example. We thought it would be helpful to sort of go a little bit further into the CAG identifiers and the functions that we mentioned. So I pulled out some of the functions from the previous slide, for example, K03332, and I've attached the CAG pathways where you can find them, and you can go to your CAG website, CAG Orthology, and find these for yourself as well. And so this is representative, these CAG pathways are representative of thousands, tens of thousands of functions that have been pieced together in maps that help you to determine what they're doing. And these are very useful for determining, oh, we want this CAG function or we want a different one. And the ones that are represented here, again, we're talking about saliva in this case. The ones that are represented here, in addition to K03332, which fructan beta fructosidase, that's an enzyme that breaks apart fructans. The next one, K03404, is a magnesium chelatase. This would be an enzyme that removes magnesium, and it could also potentially remove calcium and other metals with 2+ charge on it. The next one is K00016, which is lactate dehydrogenase. That produces lactate from pyruvate. The next one is one of the most carcinogenic functions known, K00689. It's dextrans sucrase. And this one is involved in the conversion of sucrose to fructose. The next one is K1106, tartrate succinate antiporter. So this is converting or moving these acids and exchanging them and creating a more acidic or basic environment. K03189 is a urease accessory protein. This is going to be related to breaking apart urea, which will absolutely affect the pH. And the last one is catalase, K03781, and this is related to the breakdown of hydrogen peroxide to oxygen, for example. And all of these do have roles in carcinogenic activity. If we are focused on a different score, we would use different functions, but this represents a very good example for how we go about designing scores. Next slide. What you're looking at here is the final results for pulling together specific functions, tagging the molecular data with those functions. And so in the square on the left, you can see the functions along the y-axis on the left, and on the bottom are all the samples that we are measuring. And then the box to the right are the principal component loadings, and they're represented both to the right of the big box, and they're represented numerically and amplitude in the box to the right. And so together, this is how we get the score. And what we end up doing is we take the weightings of those loadings. Again, this is principal component 1 of the data to the left. And we take those loadings and we apply them to every individual sample after this to come up with a score. Next slide. So one of the things that Viome is focused on is trying to determine what's healthy and what's not. And very oftentimes we will come up with recommendations to say, if you have a not optimal score, you should have this supplement or this food. And what I'm representing here is an example of how we might do that. This is a very rough representation. We do a bit more mathematics to this rather than just putting a red and a green line. But this is an example of where we might decide these folks would fall into the non-healthy phenotypes. These folks would fall into the healthy phenotypes. And it is approximately related to how the molecular data is sorted on the left. Next slide. This is the final slide, and this is sort of the summation of what we did in the manuscript, which I have highlighted here. We have 8 scores that we designed that are related to stool, and we wanted to demonstrate how each of these scores behave across related comorbidities. And here we've chosen the IBS spectrum. So we have IBS constipation, IBS diarrhea, and IBS mixed type. And as you can imagine, people with constipation and diarrhea might have very different microbiomes. And we actually see that represented in these scores and the odds ratios for these scores. So for example, I'll just pick out one example with methanogenesis and constipation. So this is the second panel down towards the methanogenesis pathways and constipation. When this score is not optimal, you are more likely to have constipation. Now I can go into the molecular mechanics of this. It's basically due to gas exchange. You're converting hydrogen to methane and therefore you have lower pressure. So in this case, when you have a not optimal score, you're more likely to have constipation. And that's what's represented throughout this chart here. You have a line on these panels, which is related to 1. So the closer you are to 1, the more likely you are to have no significant difference from healthy or good. And then the further you move away from 1, the more likely you are to have a not optimal score. And in this case, you can see there's almost a 2-fold increase for this odds ratios linking IBS constipation and methanogenesis. And that's it. I'm happy to answer questions.
Great. Thank you so much, everybody. We appreciate your time and your input. There are a number of questions in the chat, so I think we're going to start there. Let me myself. Let's just take the spotlight away, and let me bring myself back.
I answered a couple of
Very good as well.
questions.
Great. In the interest of the recording being complete, Mpeg-g, I'm going to ask you to go. I'm going to read out each of the questions, and
Okay.
then I'm going to ask you to answer them
Okay.
loud
Yes.
as well. Thank you. Okay, so the first question: how important is the BMI in the study? I rarely see it in omic studies, to my knowledge. I think that would be referring to the original, the Snyder study, or the Zoo study. I assume that's aimed at you, Mpeg-g.
I think it's the Viome studies providing BMI, right?
Yes, we
Maybe I'm wrong there.
Yes.
I think
Yeah,
both
I can answer
have the
some.
BMI.
So
Yeah.
BMI is important. We often see signals in our data related to BMI, and you can find publications demonstrating this at the microbiome level and the blood level. So I would definitely say it's important to some degree, depending on what you're trying to do.
The point here is the Snyder study was focused on type 2 diabetes, and the Viome study, the Viome data set, does not have hemoglobin A1C and other measurements that might tell us the diabetes status of the person. However, instead, we have BMI, which is often correlated with, it's not always true, but it has been studied and there is significant correlation between BMI and type 2 diabetes. So that's why we included that.
Okay. The next question, 16S RNA is not a simple copy gene in bacteria. Do we need to process ASV data by GCM? Those are several acronyms that I don't know. Do you know? Does that question make sense to the rest of you?
Maybe they can be more clear. What is it that they're asking?
Okay.
I mean,
So that question
as an
isn't,
assertion, it's true. It's true. 16S rRNA is not a simple copy gene in bacteria. But this really brings us to the amplicon sequence variant data, which retains this single nucleotide resolution of the 16S reads. So, I mean, this is more granular than the OTUs, the operational taxonomic units. And this really is more granular and high resolution, if that is what you were asking. But if you meant by GCN graph-based deep learning methods, if the downstream model is really meant to preserve the taxonomic or phylogenetic structure, then yes, GCNs can be helpful. Did I answer your question, or I mean, I was trying to decipher. Okay.
I mean, it looks to me like the question was answered. Hopefully, if the person who asked the question has follow-ups, they'll put them in the chat. Yes, looks like you answered it. Okay, so the next question, will we get the e-values for gene function names when they were queried from databases from NCBI or elsewhere?
This is more of a Viome question, I think, or?
Is that a question that makes sense to you, Lan or Eric?
And maybe
I'm wondering if this is asking about mapping.
Right. So I think this is about if you have a particular sequence and then you use NCBI and then you will find something and that will come back with the e-value with some the reference that it can be mapped to. So NCBI, I don't believe has the KO annotation directly. If you just paste a sequence on their UI and then you select their reference databases. Usually it's the NR, which is non-redundant refseq NR, and you could select the other collections, but those would be the genome annotation and the gene annotation. I don't believe NCBI will give you the KO annotation, that is part of our own curation process to map the genes to the function, these kind of mapping, and it's the many-to-many relationship. Many genes could be mapped to the same function, but this gene could also have a different function.
Okay. It just became more interesting, right?
Yeah, I hope that's clear enough, Nebuti. If that is not clear, please do ask us for that. We'll get back to that. Okay, next question. What is the microbiome? What do the microbiome features in track 1 inputs mean? Does that refer to the abundance table? So, can you answer that?
My answer is, as we introduced in the last webinar, and also it's in the starter notebook, you may decide to infer features in your own way. And that's why we wrote it in such a generic way, or you may decide to go the standard route of ASVs and OTUs. So we just don't want to prevent you from being very innovative and compute your own features.
So it might be the case that somebody who's participating in both challenges would have done significant data exploration, identified new features or synthesized new features, and then they could bring that into challenged.
Absolutely.
Fantastic.
Actually, I
Thank
want to
you.
add something else here. There is a standard process that now we are using from Mpeg genomics compressed data. We decompress and we have the raw reads. They're all pre-processed because we removed all the human reads, so they're all the microorganism reads. And then we have all of the downstream analysis. Now, if someone is really brave, they can start with the compressed data. And originally we discussed that would have been the primary goal, but that's a very tall order. So unless you are kind of an expert both in compression and AI, maybe that will be a much bigger challenge. That's my comment to it. Yeah.
Okay. Next question. In fact, oh, that was your. Yeah, so you've answered this in the chat already, but the question was, in fact, two, we were required to give microbiome cytokine pairs, or excuse me, microbe cytokine pairs. Question is, do we need to show the genus name in the result?
Yeah, that would be desirable. Or whatever is the lowest level in the taxonomy that you're using that you can give. I think we cannot go to species level with the 16S data, but maybe with the microbiome data we can. So.
Okay, and then from Faithful, I'm working.
The biome data. Yeah.
Too many technical terms. From Faithful, he asked, like a he from the profile picture. Faithful asks, I'm working on fact 4. My question is on the distribution of metagenomic factors in a sample. Is the taxonomic data seen to follow a particular population distribution?
Generally, yes, but I think at this level, maybe Lan has a better view based on the data you guys have analyzed at Viome.
Particular population distribution. So,
if I just want to clarify, do they mean human population or bacterial population?
That's a good question.
Let's make an assumption. There is no
right.
answer.
I was going to say that I would assume the question is about the enrichment of a certain species, microbial species, in a region of, related to certain geographic region. So people from those region might have higher abundance of a particular type of microbes because of either the environment or the dietary, all these different differences naturally occurred. So if that is the question, bacteria population per event. Oh, so let's say if you take antibiotics, then antibiotics definitely is gonna kill off a bunch of bugs. That is an event, right? And if you have an infection, and then your immune system is gonna, gonna get armed up and to fight for the infection, so that it could change the host environment for the bacteria, for the microbes. And then that might favor some species more and then favor some species less. And if you're the bed bug triggering all these immune responses, then that's gonna be, hopefully it will be killed.
So there will be different distributions, is the short answer. Per person, per
Correct,
population.
but those events are very acute. But you just from day to day, it may not be, that dramatic, right?
Mhm.
And you take
Yeah.
antibiotic infection, that's big change.
And there's a follow-up question. Do
Yeah.
some species appear much more prominent, like a normal distribution?
Normal distribution.
Is there such a thing as a normal distribution when you're talking about a microbiome
Right, in the
population?
microbiome, because in the gut, the E. coli is very abundant, and I think everybody has very high amount of E. coli. So I wouldn't say a normal distribution. That's too perfect.
Mhm.
Yeah.
So there
Probably
isn't such a thing as a
in-
normal
yeah.
microbiome distribution or some other sample.
Probably you will see
And
different types of distributions across different microorganisms. No. There will be far from like a Gaussian normal distribution, is at least my experience.
hopefully that shed some insight, Faithful. Are there any other questions from anybody else? And it's quite okay to ask in the chat, but you can also come off mute and ask the question out loud if you'd like to.
Oh, not specific distribution. I think we have seen all different kinds of shapes.
So they aren't necessarily overall patterns that you can say for that population that you had.
I don't think if this direction is going, if there's some prior, you could put some prior knowledge of that, what the data or the abundance data you're gonna get, that probably is not going to help.
All right. Seems like that is the extent of the questions here. Thank you all so much for the questions. It's great to see people engaging. I'm sure that as soon as you really start working on this challenge, you will have a lot more questions. I'm really glad to say that Naventa and some of the other experts that have worked on these challenges are going to make themselves available to answer any of those questions. So if you do have questions, we have a chat functionality within the challenge on Zindi. Please do ask your questions. Please share the insights that you are generating. If you find something interesting, I'm sure other people would like to know about it. So do feel free to share some of your analysis, some of your questions. And if you do get stuck, we definitely will have experts in the field on hand to answer some of those. Um, yeah. So that I think brings us to the end of this webinar. So all that's left is for me to say a very big thank you to Nevenka, to Lan, to Eric for your time and your expertise that you've shared with us today. Thank you also to the other technical partners for this challenge that weren't on the call. And thank you to all of you for taking time out of your no doubt busy lives to spend a little over an hour with us to talk about a really awesome set of challenges. And I wish you all very good luck on the challenge going forward. Thank you so much.
Thank you for organizing Paul, and I would like to also thank Lan and Eric and underscore that this type of data set that Viome has made available for us for this hackathon is really one of a kind. It is very rich data set, and as Lan said, 99% of the data out there is 16S. Metatranscriptome, I think Viome is really the world leader, and this is kind of a gift to humanity what we had on this call. I just want to underscore the importance of what you just heard from Lan and Eric. I think they're world experts in this area, and metatranscriptomics in the future is the way to go because it really shows you the active microbiome. I think 16S was just the start for the microbiome era. And I hope you're motivated to look into this data set. They are available to answer questions. And I think it would be very exciting if that track 5, we left it completely open. So you can think of new ways to plow through this data, to grok this data, to analyze it, to build models that no man has seen before. So I hope you engage and you bring something completely new. Just think of it as quite new and quite underexplored territory. We hope that we have breakthroughs here. I hope that at the end we have enough novelty so we can publish. We're approaching some journals to see who wants to host, but we need to see the novelty in the approaches. So just if you had any doubts, just go and reach for the stars, because I think we have some very, very important nuggets in this data that until now, very few people have been able to have access to this kind of data. And I know, as Amy says, this is a brilliant crowd and very motivated. So I have very high trust that very novel things will be discovered and proposed in these challenges. Thank you. Thank you again.
Thanks, Nevenka. We're looking forward to it.
Thank you.
Thanks, everybody.
Bye.
Yeah.